and your different mic and camera has been disconnected, so you can only focus on the different presentation that we will have. And you are invited to use the Q&A box should you have questions during the different presentation. And for those who want to directly ask their question at the end of the different presentation, we have a dedicated Q&A session where you will be invited to directly ask your question. So you can raise your hands and then we will open your mic. And also, today we're having our third module in our health journalism bootcamp series. And after understanding malaria and also talking about gene drive and talking about genetics and gene drive technology, today in our webinar, we'll be talking about the regulation surrounding the development of gene drive technologies in Africa. How or in which setting, according to which framework we can work on developing gene drive technologies. Today we have experts with us to just let us know how this can be done following the different regulations. Also, we'll be discussing the ecological studies that have been undertaken by Target Malaria in order to see what they're doing with gene drive regarding the mosquitoes elimination or replacement of the population. What are the benefits or what are the risks? This will be discussed during today's webinar. And as I said during the introduction, I will be co-moderating the session with Divine Jokoto, who is the stakeholder engagement and communication leads of Target Malaria. Without further words, I will pass the floor on to you, Divine, for your introduction. Thank you.
Thank you, Eric, for the introduction and for the background rules. First of all, I'd like to give some context about Target Malaria before we go into the presentations. Target Malaria is a not-for-profit research consortium. The aim is to develop and share new, cost-effective and sustainable technologies to modify mosquitoes and reduce malaria transmission. Our vision is to contribute to a world free of malaria. Just a moment. I am delighted to introduce our first speaker, Dr. Charles Mugoya. Dr. Charles Mugoya is the Regulatory Affairs Manager for Africa at Target Malaria. Mugoya is an entomologist by training. For the last 25 years, he has been involved in capacity building initiatives in biotechnology and biosafety in Uganda and other countries in Eastern and Southern African region. Prior to joining Target Malaria in 2021, he served as the National Biosafety Committee Chairperson in Uganda, where he led and oversaw the regulatory process for biosafety. I present to you Dr. Charles Mugoya, who will go into his presentations now. Thank you. Dr. Charles Mugoya.
Thank you very much, Divine. It's a great pleasure for me to make this presentation at this Health Journalism Bootcamp. And I'll be speaking about a subject that I'm extremely passionate about, namely the regulatory environment, pathway and risk assessments that accompany the work that we are doing in Target Malaria to deliver on the outcome of reducing malaria. And in my presentation, I will be focusing on the regulatory environment. I will also focus on the pathways and risk assessment as the requirements that are needed to deliver successfully on this technology in terms of compliance with the established norms, regulations, and standards. So I don't know how we can do this, but can you advance the slides forward for me? So I'll start by saying that the first genetically modified Anopheles mosquitoes were modified at labs at Imperial College London about 23 years ago. So it is quite some time since we started this journey. And I just want to say that modified organisms or genetically modified organisms are typically organisms that have been modified from the original strains, but in this case, we have used laboratory insect strains to do the modifications. So as it were, we have developed at this point in time what we would refer to as a first generation genetically modified strains. And the purpose of doing this first generation is really to develop an environment for learning about the attributes of this genetic modification. And we want to study also the biology of the mosquitoes. And therefore, we are doing that using those modified organisms, but they do not carry the gene drive technology. And over time, we have been able to work with and collaborate with the African partners, because this is a problem for Africa. Malaria is a problem for Africa, but we had to make a start. So we chose Burkina Faso and Mali as the countries where we would first test our concept and under laboratory conditions, which we call contained use. And we secured the necessary regulatory approvals to be able to do that testing, to facilitate the testing in those two countries, in Burkina Faso and Mali. Next slide. So the pathway that leads to a regulatory environment is basically a very simple pathway. There are three pillars of this pathway that lead us to something that we desire. And that something that we desire is the product that we are aiming at, a product that will go in the field, that will go into the environment, that will be able to suppress the population of malaria-carrying mosquitoes. So we start off with the discovery. And the discovery is a very important aspect of regulatory environment, because it's where everything else starts. Sorry about that. That's where everything else starts, at the discovery of the concept. And then in the process of discovery, the first step is we have to do the development aspects of the research. And those aspects of the research are very, very important for regulatory startup points. So it involves the proof of concept, it involves laboratory work, it involves cage studies. And that informs the next stage. The next stage is it acceptable? And therefore we do carry out scaled evaluation, in which we do several impact analyses, which are both at laboratory level and environmental level. And the idea is we were securing public acceptance. And once we secure public acceptance, then we go to the last stage, which is the implementation. And the implementation is made up of two elements mainly. How do we deliver on the product that we are promised? And how do we interact with the public so that we can be able to work with them in the delivery plans of our project, of our product. Next slide. So the state of play as of now is something that we are working with, and it involves first three key elements which guide the process. So we have the international binding agreements. For example, the Cartagena Protocol is one of the most important internationally binding agreements that we focus on. And this agreement sets the focus, it sets the scope of things that we do as regulators. And it gives us ample guidance how to proceed at regional level and at national level. And then there is also guidance at regional level, which is provided by the context of the environment where you are operating. For example, the African Union gives some guidance on how we need to proceed with the technology. And then we have the national laws and regulations, and the national laws and regulations set the context in which we are working at local level. So there are policies, then there are regulations that countries set in the context of the national biosafety framework. And then as we do that at all the three levels, we have the public. There's the international public, and many of you are aware about the Conference of Parties to the Cartagena Protocol, where parties, which are government entities and NGOs and other players, come together to discuss the elements of the technology, discuss the safety, and they also discuss the benefits and risks. So that is the public at international level. Then we have the public at regional level, and we also have the public at national level. Now, the idea behind the involvement of all these stakeholders is to build public trust, but also to ensure that we do not leave any stone unturned as far as safety is concerned. So those three pillars, as you can see from this diagram, they interact with each other, and you cannot remove one and not affect the other. So basically what happens is that the countries where we are working with have adopted national frameworks for genetically modified organisms. And that framework guides the process in which we are working. And that framework is made up of the laws, the policies, but also institutions that have been set up to facilitate the administrative processing of the requirements for the implementation of the technology. Now, our regulatory approach therefore considers that gene-drive development is a practice that is regulated. So you cannot do gene-drive development, for example, as we are doing it today, without any regard to a regulation. And then secondly, we have made investments in a phased program development. So what does this mean? It means that we want to approach the development pathway in phases. And for target malaria, we from the very beginning, we said we shall have a phase that is startup phase, proof of concept, laboratory studies, and we chose the model of using sterile male mosquitoes as a learning phase to test the technology. And then we went into the preparations for paternal male bias, that is male mosquitoes, predominantly male mosquitoes, as our product that we use for testing, again, the pathway for gene drive. And then eventually we shall have the gene drive going into the environment. So this investment in phased program development. We understand it as a best practice so that we collect systematically all the information, all the knowledge about the biology of the genetically modified mosquito as we go along. And then thirdly, and most importantly, we have adopted a principle of case-to-case assessment. And this is a powerful principle because, as you understand, no biological organisms are the same. Not all the biological organisms are the same. And therefore, even the mosquito species that we are dealing with are not the same. So we are going with this principle and learning how we have chosen to use the target organisms, which are mosquitoes that are spreading malaria. And in this case, we are using them to be able to understand and be able to deliver on the promise of reducing those target organisms, which are the malaria-carrying mosquitoes in the environment. And that case may not apply to other cases that we have had previously. And it will be important also to understand that this is the first time, if we succeed, this will be the first time that we are able to deploy a genetically modified mosquito, which is attributes of gene drive into the environment. Next slide. So our current regulatory environment in Africa is of a mixed nature, but we are all aiming at the same end product at the end of the day. So each country which is participating in the project engages regularly with the national regulatory bodies and in a co-development fashion. That is, we share information, but we also share the results of the risk assessment that is coming out of the research. So for us, therefore, it is very important that a national biosafety framework must be available. And countries that have this biosafety framework are the countries that we're working with currently. And each country has its own biosafety authority, and that's the authority that's in charge of regulating this activity. And because the countries are formatted differently, this authority sometimes is in the Ministry of Environment, in other countries in the President's office, in other countries it's in the Ministry of Science and Technology. So it depends on which country you are dealing with, but the most important thing is that it is the authority, the biosafety authority, that is in charge of regulating the activity in a particular country. Now, in some partner countries, a lot of this regulation is also done by the environment-based bodies. And we have the National Environment Management Authorities that are doing the work of regulating the technology in the country. So when we go into the country, the first thing that we need to do is to understand what are the institutional arrangements and how is the regulation of the technology or the genetically modified organism done. And then we don't work in an insular environment. We also work with the African Union, which is the technical arm of the African Union NEPAD organization in administering the regulatory oversight over this technology. And I need here to mention that the African Union has been at the forefront in identifying technologies that will reduce the burden of malaria in Africa. And the African Union Commission, the Secretariat of the African Union, has published a document called the Gene Drive document to support the understanding of how this technology can be useful in reducing malaria in African countries. Next slide. So basically the regulatory approach, as I have highlighted, encompasses the policy-related aspects, which includes the laws, the guidance, resources, the regulations, which are proportionate to the issue at hand, the issues on risks and benefits, then in general, the enabling environment. Those are encapsulated under the policy arena. But then without the evidence, then the policy cannot work alone. So we have to adduce evidence. And this evidence must be sound and scientific evidence that also includes community inputs. Now, in addition, this evidence must be supported by what we call plausible pathways to harm. And now that is, we interrogate every aspect of perception or opinion about the harms that can be caused by, for us, releasing our technology. And therefore we must base our decisions on the premise that we have done research and we have identified possible pathways to harm, and we have followed those pathways. We have developed hypotheses, and we understand that these pathways will even if they were completed, they will not lead to the harm that we anticipate. And if they lead to harm, then we have to mitigate. So that is evidence, and that evidence is very important in the regulatory process. So we cannot, for example, achieve our end objectives of reducing malaria using this technology without the analysis of those pathways, that possible pathways that can lead to harm. And then we have to test and verify that the risk and impact assessment gives us comfort that if we release this technology, we will not have any risks in the environment or to human health. And therefore, there are thorough studies that are done regarding environmental and social impact analysis that give us the evidence that we need to present to decision makers, the national biosafety focal points, the national biosafety authorities, that the technology that we are going to bring on board is safe and sound. And then the other pillar is compliance, because in the process of implementing the technology, the countries where we work give us certain benchmarks. They direct us to follow certain processes. And therefore, when we submit our application for the release of these organisms in the country or testing, we must adhere to the regulatory terms and conditions attached. And those are provided by the laws. They are provided in the standard operating procedures, in the manuals, and in the regulations that the country has put forward. And then we then work under a permit or license, because the assessment from the evidence will give us a certain a scope in which we have to work, and all that is given under a permit or environment. So it means that under regulations, we only work where we have been permitted. And therefore, we cannot work in countries where we haven't received permits. So it limits our scope for testing, but the end product is that we generate information that can be used for the entire continent. Next slide. So I've talked about the evidence, and the evidence is adduced through risk assessment, risk management, and risk communication. Next slide. So these three elements, the risk assessment, the risk management, and risk communication, actually define the scope of risk analysis that we undertake when we get involved at country level. And this risk analysis is extremely important. It is a very important part of the regulatory process because it defines the success of the technology. People will be comfortable when they know that the risks, there are no risks, or the risks are very low. But if the risks are very high, then the decision will be taken that this technology should not be released into the environment. So risk analysis, therefore, is a very central element to decision-making from stakeholders. And whether to go into this technology or not will depend on the risks that have been identified. So, as I've said, it comprises 3 elements: risk assessment, risk management, and risk communication. And at Target Malaria, we have dedicated teams that are working on these 3 components. And they are able to identify the potential harms that may result. They're able to identify the concerns of stakeholders. They're able to analyze the concerns and even identify those concerns that warrant further characterization. And additionally, we carry out internal risk assessment where we test our parameters, the hazards and exposure characterization and risk characterization. And all this is done to support external, all this is done with the support of the external independent risk assessment, because we cannot rely solely on the internal process. We also need some input from the external inputs from the external bodies that are with expertise in risk assessment. And above all, we identify the potential management strategies where risks have been identified. And therefore, the end of the day, we're able to design a product, and also we use operations that reduce risk. So all this is done in a very systematic manner. And we have hired and worked with organizations who also inform us on the project regulatory submissions that we prepare for the national authorities. And these are independent bodies, and their input is to ensure that we have not left anything out there which can contribute to the success of the risk assessment and the risk management process. Next slide. So, so the process, as I've described, is therefore a lengthy process, but a necessary process, starting with the problem formulation, going into hazard exposure characterization, eventually leading us into risk characterization, where we are able now to start the engagement with stakeholders to understand the magnitude of the risk, the benefits of the risks, and the management strategies in case the risk exists, but it is mitigatable. And then finally, we reach a risk conclusion, and that risk conclusion defines the decision with which national regulatory authorities go with. So this process from problem formulation to risk conclusion is extremely critical in the process of risk assessment. And it involves stakeholders, involves institutions that are mandated with this function in every country. Next slide. So we have had a lot of experience over the years since the year 2000, and we have also had guidance in the development pathway of our gene drive insect, and there's a lot of guidance that we have received from various bodies, and in a nutshell, the guidance that we have received gives us confidence that we can move forward and do the testing for gene drive in Africa. And so far, we know that the gene drive can be addressed by the current conventional guidance for genetically modified organisms. this is a very important aspect, because initially there were some opinions that we really needed a new format of guidance from that of genetically modified insects. But as of now, I think there's ample evidence that suggests that the current guidance for genetically modified organisms can also be applicable to bear on the gene-drive insects. And then secondly, we have affirmed that we can follow the case-by-case, stepwise process as a best practice. And this is also going to help us very much in understanding the process as we go by, so that one phase guides the formulation of the next phase up to the time when we release the final product that is flawless, that has the benefits that we anticipate. And we also now understand that this process applies the same mechanisms. The gene-drive process applies the same mechanisms for the preferential inheritance of a transgenic construct, like any other GM construct, including the ones that are for potential, that have potential for population replacement, like in the case of mosquitoes. And that the guidance that we have on gene-drive is that it has the same similarities as the ecological issues that we had from the experience of other suppression methods, like the sterile insect technique, biocontrol mechanisms. So it is the same ecological issues that we interrogate, that we benchmark with. And also the similar molecular similarities in the analysis and similar methods for detection. So that gives us confidence that the current guidance is as good. And it also needs control systems as comparators. So this is the same as any other GM organism. And therefore, what we need is maybe to do additional fine tuning. Now, a lot of this guidance for gene-drive is also available in literature. We have the guidance from EFSA, that is the European Food Safety Agency, and also from WHO, which have given similar kind of guidance. They don't see any differences between a GM organism and a gene-drive organism. So that guidance has given us the confidence to move ahead. Next slide. So this risk formulation for suppression of gene-drive publication was a publication that came out of the experiences from the pathways process, where we started with conducting workshops. This was followed by stakeholder technical consultations, and it helped us to formulate the current pathway that we have today. And this particular publications contains those plausible pathways to potential harm via problem formulation. And it was done specifically for investigational releases of the genetically modified population suppression of gene-drive to control human malaria vector, Anopheles gambiae. So this publication is a key publication for us, and we are following through. There were about 46 pathways that were developed, and we have followed through all those pathways in order to inform all the decisions that we are making at country level. Next slide. So there's a lot of learning that has come around as we go forward. One of the key issues that we have learned in this process for risk assessment is that additional guidance for environmental risk assessment in particular requires participation of a broad range of stakeholders. And we have also gotten to understand that the definition of target organism for gene drive applications involving species complexes require more nuanced consideration than other GMO applications. So this is something that is beginning to come up, and we are giving it due attention. We also have understood that environmental risk assessment should use problem formulation approach to address specific operational protection goals. And this is exactly what we are following through in the countries where we are currently working. And the engagement, specifically the engagement in problem formulation, has to include several actors. And the actors should also have broad expertise, because we value the expertise that is brought on board by different disciplines that are involved in the technology. We have also tested our strength in terms of possible scenarios, and we are of the opinion that a realistic worst-case scenario should be considered when testing risk hypothesis. Why is this? It's because one should anticipate anything to happen as a worst-case scenario, and therefore we value the worst-case scenarios, because then it tests the decision that we are making. Because at the end of the day, we are weighing these technologies for benefits against risks. So the worst case scenario gives us a scope in which to exercise our imaginations. We also understand that a range of comparators are needed and should be considered, especially in the environmental risk assessment. And this is extremely critical, because in order to contextualize the risks, one needs to really test against different organisms that are available in the environment. And then the other learning that we have received from risk assessment process is that we actually need in the analysis of data, we really need a mix of both the probabilistic and qualitative analysis, because these two go hand in hand. We cannot just use quantitative means in the risk analysis. We also need the extrapolation of what our imaginations of the probable risks that may occur. And then we have to weigh them against the levels of uncertainty and the nature of the risks. And then we also understand that we needed to use concepts of ecological interaction, networks, to assess impacts on dynamic properties of the basis, on the basis of biological considerations and stakeholder values. And lastly, we understand that the engagement in the environmental risk assessment should complement actually related impact assessments, should also link up with risk governance of frameworks and the national policy contexts. These are very important, and they are supposed to be taken together so that we can now get a decision that appeals to people, appeals to the public when it comes to the question of risk assessment undertaken. Next slide. So I have talked, I've explained the importance of independent risk assessment. Next slide. And at the end of it all, there has to be some components of risk management. And risk management is a systematic process to bring the risk to an acceptable level, because there could be some risks, but it is mitigatable. And therefore, we carry out logical analysis to identify pathways to harm and the information that we need to ensure those risks are acceptable, and the plans that can be used to change or deal with unacceptable risks. I'm sure everybody understands that in life situations, sometimes you have a risk that is not acceptable, but then it can be mitigated or reduced in impact and be tolerated. So we try to avoid risks by ensuring a careful design of products and operations needed by the project. But we also plan operations very diligently, and we do this to minimize exposure and also reduce the risks. And then we, in addition to that, we make contingency plans wherever a possible, feasible risk is anticipated. And then we monitor and mitigate the risks if they arise. So we have thought through all these processes for genetically modified mosquitoes, and we have covered all these areas for risk management. We have planned, we have carried out logical analysis of impact pathways. We have put in measures to avoid risks through the design of our experimental products. We have planned all the operations to minimize exposure and reduce risks. And we have made contingency plans. And all these are packaged in standard operating procedures that we have developed as a project. Next slide. So I alluded to the international guidance documents, and in the next 5 minutes or so, I just wanted to go through some of these international guidance documents and relate them to how they are helping us. So we have the CBD at international. The CBD is the Convention on Biological Diversity, and then we have the Cartagena Protocol on Biosafety. Then we have the Nagoya Protocol on access and genetic resources, and the fair and equitable sharing of benefits. And then we also have the Nagoya Kuala Lumpur Supplementary Protocol on liability and redress. So these key instruments talk to different things, but at the end of the day, they guide the way we conduct our planning for genetically modified mosquitoes and gene-derived mosquitoes. So the CBD has provisions for the conservation of biological diversity, the fair and the equitable sharing of benefits, and it also contains provisions that help us to see where the benefits are and risks, and it lays down the process which has resulted to the Cartagena Protocol, which defines the risk assessment measures. And within the risk assessment measures, there was a Nagoya Protocol, which spelled out how you share the benefits. We have done that through what we call the material transfer agreements. Then the Nagoya Kuala Lumpur talks about damage to the environment in case any technology that we are using will give those damages, and it spells out regimes for liability. In other words, it spells out our responsibilities, especially in terms when it comes to damage to the environment, and it gives confidence actually that we are operating under a regulated environment. Then there are other guidance documents like the WHO guidance document, which helps us to frame the testing of the genetically modified organisms. There's the European Food Safety Agency, which has given us the tools for example, detection of the genetically modified organisms in the environment, and also among the species themselves. Then we have the National Academy of Science, Engineering and Medicine, which has published a landmark publication called Gene Drives on the Horizon. And this one gives us a whole range of best practices that can be used to engineer and get genetically modified mosquito that can control the malaria problem and challenges. Then we have the OECD, the Organization for Economic Development and Cooperation, which has developed consensus biological documents. These are documents that speak about the biology of different organisms that we are using. It has just developed a consensus document for Anopheles gambiae. And there's also another one for Anopheles aegypti. So these consensus documents give us a lot of rich resource to pick best practices from as we develop our technology. Next slide. And then we have, hello.
Yeah, sorry to interrupt, Dr. Mugoya, just to let you know that you have 7 minutes for your presentation.
Right. I think I'll finish within those 7 minutes.
Thank you.
Thank you. So next slide. So we also have African Union, and the role of African Union is to guide the orderly conduct of research. And the African Union, as you may know, has its structures. I'll just mention a few of them. It has the Secretariat, it has the Ministerial Council for Science and Technology, it has the Agency for Development called the AUDA-NEPAD, and it also has the African Biosafety Network of Expertise, and then it has the African High-Level Panel for Emerging Technologies, and then the African Center for Disease Control and Prevention, the CDC. Now, these structures are extremely important for our work, and I'll single out the African Union High-Level Panel for Emerging Technologies. This is a think tank for the technology that we are currently addressing, the gene drive technology. It has given us the sort of a scope in which to work. It highlights the possibilities. It highlights the potential. It also clarifies on the pathway that we need to use. And then, of course, there's the African Center for Disease Control and Prevention that has started building facilities that can be used as labs for scaling up any technology that we have. And then the African Union also has governance instruments. These are high-level policy instruments that are helping us to formulate the interventions that we need to make in Africa. It has the Agenda 2063, which has 7 pillars. And when we look at pillar 1, 2, and 3, it addresses technologies, it addresses capacity building, it addresses the issues of health in Africa to spur prosperity. So the Agenda 2063 is also a very good document. Then we have the STISA, the Science, Technology, and Innovation Strategy 2024. We have the African Model Law, and we have the gene drives for malaria control and elimination. Next slide. And then I just wanted to touch on the East African community because that's the region where we are working currently in Uganda. The East African community has made several guidance instruments that are helping us to articulate the requirements for gene drive. So we have the STI policy, Science, Technology, and Innovation policy. We have the East African Community Protocol on Environment and Natural Resource Management. We have the Regional Bioeconomy Strategy, and we have the Harmonized Policy Framework for East African Community. And then we have the East African Intellectual Property Policy. And all these instruments touch on the subject of the needs and the requirements for genetically modified mosquitoes. Next slide. And then we have at national level, and I'll cite 2 countries here, Tanzania and Kenya, and Tanzania and Uganda, because these are the countries where currently they're developing gene-derived mosquitoes. So we have government bodies that are involved, that we consult with when we are developing the plans for testing and deployment of gene-drive, starting from the Office of the Prime Minister, the Ministry of Science, the Ministry of Health, the Ministry of Environment. We have the National Biosafety Committee, and then we have the institutional biosafety committees. So Uganda has quite a wide range of institutions that we are currently engaging in order to safely test this technology. Next slide. And then we have, in the case of Uganda, apart from the institutions that I've highlighted, we also have policy instruments. We have the National Biosafety Framework, we have the Science and Technology Innovation Policy, we have the Health Policy, we have the National Biosafety Committee, Standard Operating Procedures, and others. On all these policy instruments guide orderly interventions when we are planning the testing and implementation of the gene-drive. And then we also have those technical guidance documents. These come as standard operating procedures, they come as manuals, they come as guidance resources to use when we are trying to navigate the regulatory landscape, as it were. Next slide. And I would not leave this talk without talking about the key legislative instruments. It is because of the various nuanced involvement of different laws, we rely on different instruments in order to test the technology. And currently, we do not have in Uganda the Genetic Engineering Act, but then we have some other laws that can help us to do the research and the contained use in the laboratory. And that is delivered through the Uganda National Council of Science and Technology Act. But we also have other acts like the Public Health Act, the National Public Health Laboratories Act, Occupational Safety and Health Act, and others, National Environment Management Act, and then National Environment Management Regulations that spell out the environmental and social impact requirements, and many others. So all these regulations are consulted when we are mounting activities that are relevant to the project. Next slide. So we have Tanzania also. I think it is almost the same format like Uganda. They have different government structures and institutions, starting from the Office of the Vice President. Then you have the Ministry for Environment, Ministry for Science and Technology, and they also have committees, National Biosafety Advisory Committee, National Biosafety Committee. They have institutional biosafety committees. These are governance mechanisms for addressing the requirements of the applications for the deployment of the technology. Next slide. And Tanzania as well has policy documents. They have the National Biosafety Framework. They have the Biotechnology Policy. They have the Institutional Biosafety Committee Manual. They have Public Engagement in Decision-Making Process Manual. And all these actually help the scientists in the laboratories to be able to define their regulatory pathway and be able to comply with the laws that are necessary. Next slide. And the laws in Tanzania are mainly three: mainly Environmental Management Act, Environmental Management Regulations, and Environmental Biosafety Regulations. Next slide. So, having said that, I wanted to just bring this talk to focus a little bit on what could be a possible role of a regional outfit be, because at the end of the day, when we are talking about regulations, and how it impacts the delivery of the technology. The technology that we are talking about, the gene drive, has a regional implication. It has transboundary implication. And therefore, you cannot, as a country, work as a silo. You need to involve this and bring it to a regional level. So I'm suggesting, I've put up a proposal that we need to work as a region. And therefore, we require a regional biosafety framework. We require some kind of regional standards in terms of documentations to facilitate, harmonize the assessment of the rollout of genetically modified organisms research and product. We also need to harmonize regulatory processes, because regulatory process will define the extent to which you deploy this technology. And at the end of the day, when we have got a product that we are able to release into the environment, we therefore need a distribution mechanism. And that, if that mechanism is regionally constructed, then it will give us more confidence to even look at aspects of monitoring at regional level and look at the whole essence of surveillance at regional level instead of looking at national level. And then I think it is very important for us to promote capacity building as a region rather than as a country, because the product that we're doing research on is a regional public good. And therefore, if we promoted that capacity in the area of risk assessment, in the area of risk management, the risk communication at a broader range, then we'll be able to address some of those issues of deployment of the technology that will make it successful once it is rolled out. Next slide. So I think with those, I'm very positive that when we work together as a team, we will be able to succeed, and we will be able to reach the end goals. And the regulatory function is going to play a very, very important aspect of this, because this is where we are going to get the policies, the decision makers, and the implementers, the scientists, all working as a team. So I want to acknowledge the 11 institutions that have played big role in the development of these plans for this technology. We have more than 200 experts who are working with us. We have a whole large section of communities and stakeholders who are working with us on different aspects of this technology. So I thank you very much. And we want to acknowledge all the organizations that have worked with us. Thank you.
Eric, you are on mute. We cannot hear you.
Oh, sorry, Morgan. Yeah, I was just thanking Dr. Mugoya for his presentation and for sharing with us these key information. As takeaway, we can just note that in order to develop gene-drive mosquitoes, we need to have regulation, and there are regulations that exist at international, regional, and national level. And then Dr. Mugoya shared with us the case of Uganda and Tanzania that have regulation in place, but also regulation at the regional for the African Union, but also regulation from the WHO and other institutions. Thank you very much, Dr. Shal, for your presentation. And thank you also to our attendants who are sharing questions with us in the Q&A box. we'll just come back to this question when we'll be answering the question. I will pass the floor on to Davin for the introduction of Dr. Fred for the second presentation. Thank you.
Yeah, thank you, Eric, for that wrap up. I'm delighted again to introduce our second speaker, Dr. Fred Aboagye-Antwi. Dr. Fred Aboagye-Antwi is the principal investigator for target malaria in Ghana. He's also a medical entomologist at the Department of Animal Biology and Conservation Sciences, a senior lecturer within the School of Biological Sciences at the University of Ghana. He has over 20 years experience in research on parasitic infections. He joined Target Malaria in 2018 and has been investigating the ecological consequences of reducing the population of Anopheles gambiae, Anopheles mosquitoes in malaria endemic regions. In addition, he's working on optimizing protocols for rearing, transporting, and releasing male Anopheles mosquitoes. I introduce to you Dr. Fred Aboagye-Antwi, who will be making his presentations shortly. Thank you.
Yeah, thank you very much, Divine, and good afternoon to everyone. So I'll be talking about the work that we are doing here in Ghana, and I hope I will build on what Charles spoke about earlier in the context of what we are doing here in Ghana. Next slide. So yes, as you may be aware, malaria is transmitted by mosquitoes. And so far, presently, about 3,500 different species of mosquitoes have been identified, of which 837 are in Africa. And of these, it's just about 40 species that transmits malaria. But when it comes to sub-Saharan Africa, the most important malaria vector to us are members of the Anopheles gambiae species complex. And there are 3 of these that are responsible for over 90% of all the transmissions. And these are the ones that Target Malaria is focusing on. And these are the Anopheles gambiae, Anopheles coluzi, and Anopheles arabiensis. They are all members of the Anopheles gambiae complex. And just as Charles said, Target Malaria is using the phased approach. So we are progressing in a stepwise phase manner. And all that we're doing has to be diligently and carefully worked out. Next slide. And so presently, the state of progress of the work of Target Malaria is such that if you take countries like Burkina and Mali, in Burkina, non-gene drive modified sterile male mosquitoes were imported in 2016, and they were used for contained use experiments, which was ended in July in 2018. And in July 2019, the first small-scale release of modified mosquitoes. These were not gene-drive mosquitoes, but modified mosquitoes was carried out in Burkina Faso. And in Mali, they imported the first modified mosquitoes into the country in 2019. And they also concluded contained use experiments in July of 2022. And in that same year, the work in Mali was concluded. And in Burkina and Uganda, there's another kind of work that's ongoing where non-gene drive modified but male bias mosquitoes are being worked on. And the current phase of the work is such that work is going on in the lab on these. And these mosquitoes were imported into Burkina in March 2022. And as at August 2022, Uganda has submitted a dossier for contained use experiment using these non-gene drive genetically modified male bias mosquitoes. In Ghana, our work is on the ecology of the mosquitoes and also on rearing studies. And just as Charles mentioned, the regulatory goals is to protect the environment or protect biodiversity and also human health and animal health and then water quality as well. And it is in that context that in Ghana, we are working on the ecology of these mosquitoes. And I'll give details of our work here pretty soon. So with a gene drive approach being used by Target Malaria, these are the male bias and the female fertility approaches. And they are still being developed in the lab and they are still being tested in small and large cages. Next slide, please. So I will be talking about the work that is going on in Ghana in support of the regulatory aspect of gene drives. Next slide, please. So our work here involves establishing an ecological observatory to study malaria transmitting mosquitoes and their impact on the environment in general. Next slide, please. And so this work is influenced by the fact that if you take a mosquito, it has an adult niche in the sense that the adult mosquito flies around, whilst the larvae or the immature stages is aquatic, or we will say that the immature mosquito develops in water. And then potentially, these mosquitoes could be pollinators in the sense that they feed on plant juices. So we want to better understand how the mosquitoes interact. First, as adults in the adult habitat, as they fly, how do they interact with other organisms? And then in the water, where the young ones develop, we also want to find out how they interact with other organisms in the water. Do some organisms feed on them? And if they do, what will happen to those organisms if those mosquitoes are no more? And then we also want to understand if these mosquitoes are important in any way in pollinating plants, because pollination is very, very important, not only for the plants themselves, but for all other organisms that may be dependent on those plants. Next slide, please. And so our studies here in Ghana is looking at how the mosquitoes interact within the adult niche And so we will ask the question, what feeds on the mosquitoes? Do we have birds that feeds on the mosquitoes? Do we have other insects that feeds on the mosquitoes? Do we have lizards or bats that feed on the mosquitoes? And in the aquatic environment where the young ones are, do we also have other organisms that are feeding on the mosquitoes? And do they interact? How do they interact with these organisms and other organisms within that environment? And also, we also want to understand if these, as I said, if these mosquitoes are involved in any pollination at all. And the question that we ask, please, next slide. The questions that we ask is that what will happen to all these other organisms, to the plants that potentially mosquitoes will pollinate, and also what will happen to those animals that feed on the mosquitoes and which interact with the mosquitoes when the mosquitoes are no more, when we've controlled them such that they are no more, what could potentially happen to all these organisms. And then we also want to understand how to rear these mosquitoes and transport them and release them in such a way that they will remain strong enough to be able to withstand all the conditions, the environmental conditions out there, the high temperatures, the humidity, and so on. So these are the kind of questions directing our research here in Ghana. Next slide. And so the main question is, once the mosquito is no more, once the mosquito that transmits the malaria, specifically Anopheles gambiae, is no more, what will happen to all these organisms? The insects that feed on the adult mosquito, the birds that feed on the adult mosquito, the lizards, the bats, what could potentially happen to them? Are they going to die out, or nothing at all will happen to them? They will find alternative sources of food. We pose the same question when we come to the aquatic environment or to the water where we have the immature mosquitoes developing. And then if they are involved in any pollination at all, what will happen if we significantly reduce the population of the Anopheles gambiae? Next slide, please. And so the main objective of the ecological observatory study that is being carried out here in Ghana in support of the GeneDrive work is to determine the ecological impact of the removal or the ecological impact of the significant suppression of the malaria mosquito, the Anopheles gambiae. And so our specific objectives are, one, to identify and characterize the niches and the interactions between organisms within those niches and the mosquito. And our second specific objective is to unravel the role of the malaria mosquito in the terrestrial food web. This is the adult niche. So when we talk about the adult niche, where the adult kind of dwells, how the adult interacts, and also to build food networks of this kind of interaction. And if they provide any ecological services, these are, this is our second objective. And our third objective is to model, mathematically model the impact of significant suppression of Anopheles gambiae on the overall health of the ecosystem. So we want to better understand what could potentially happen if the malaria mosquito numbers significantly drops, is it going to impact on the health of the ecosystem or not at all? So these objectives are what are guiding our work here in Ghana. Next slide, please. And so here we started with kind of documenting what are the organisms in the niches of the mosquito. And so we started with the terrestrial habitats, the adult niche. So these, what we see here are some of the traps that we deployed to collect insects or invertebrates that interact in the terrestrial habitats of the mosquito. And so here we see the moth heat trap, which is deployed in the night to collect potential predators of mosquitoes that are invertebrates and do come out in the night. And with this, down here, we have the malaise trap, which has the white top and the black net, which is to trap insects, flying insects during the daytime. And we have the CDC light trap for trapping blood feeding insects and mosquitoes in particular. And then we have the yellow pant traps for trapping crawling insects that could potentially be feeding on mosquitoes. So these are some of the traps we deployed to collect our samples to better understand the interactions between these organisms and the mosquitoes. Next slide, please. And so after collecting all these samples, we build what is known as the network. And what we see here is just an example of such network. You could see a bird towards the bottom here, and you could see a couple of lines from that bird linking to other organisms within the network. And this shows that if that particular bird is not there, all those lines, the linkage between that organism, the bird and other organisms will be broken, which means that the bird could be playing an important role within that particular network. So what do we do with the samples that we collect? When we collect them, we build, first of all, a library of them. Using DNA barcoding, we build a library, and we also kind of do dietary analysis. What the organisms have eaten, we do metagenomics on them to get to know if they've eaten any mosquitoes or not. So after doing that, we build a network and we use mathematical models to predict what could potentially happen if we significantly reduce the numbers of the Anopheles mosquito or the malaria mosquito. Next slide, please. And so when it comes to the ecological impact, our goal is the suppression of malaria, not to make the malaria mosquito be extinct per se, but our main goal is to significantly suppress malaria and, if possible, eliminate malaria. And what I'll take us back to one of the very first slides where we talked about the fact that as of now, there are some 3,500 species of mosquitoes that are known, of which there's 837 in Africa. The Anopheles gambiae is just one of such species. It's just a very small proportion of the total number of mosquitoes, the total species of mosquitoes. So we do not believe that significantly, if we reduce their numbers, it could impact on the ecosystem. But what we are doing is to try and scientifically prove that that is the case. And one thing we note also is that these mosquitoes have a strong association with humans, and they are not keystone species. When we say they are not keystone species, it means when you take them out or you significantly reduce their numbers, they are not going to impact in any way on the ecosystem. They are also not known to be specialist pollinators, and that's the largest ecological effect that we expect when it comes to reducing their numbers significantly is a reduction in malaria and possible elimination of malaria. And we've seen a similar thing in places where bed nets have been extensively used. Next slide, please. And so here in this slide, we are showing some data that kind of shows the numbers of the malaria mosquito Anopheles gambiae over the years in response to the use of bed nets. And you will see that in the latter years, 2007, 2008, 2009, their numbers significantly drops. But these drops in numbers have not been associated with any ecological effect or biodiversity concerns, and hence we do not think that significantly reducing Anopheles gambiae numbers or malaria mosquito numbers can affect or could affect or could have significant impact on the ecosystem. But our work as scientists is to provide evidence and data in support of that. Next slide, please. So, in working with these modified mosquitoes, we also have to ensure very high standards, the utmost standards of biosafety. And I'll be talking of some of the activities being carried out with respect to this in our subsequent slides. Next slide, please. And so when it comes to the first phase of the work of target malaria in Africa, it involved the building of infrastructure and the building of capacity, as well as expanding on the expertise of persons that will be working on the mosquitoes to ensure that they handle the modified mosquitoes safely. And this involved the construction of new facilities and the upgrading of existing ones. And we will go through this in our subsequent slides. And it also involved the developing of SOPs or standard operating procedures that ensures that risk is managed efficiently. And so it involved containment and monitoring and reporting approaches that ensures that risk is managed efficiently. And when it comes to the building of capacity, personnel are trained on how to handle materials and how to handle the modified mosquitoes, and also how to manage the facilities efficiently, and also to ensure good bookkeeping in terms of record keeping and reporting. And we also, within Target Malaria, have an audit team that audits and conducts an internal audit of all the activities of the team members within the project. Next slide, please. And so with the construction of facilities and upgrading of existing ones in Burkina Faso, the IRSS, which is the institution that we work with in Bobo Dioulasso, carried out an extension of its existing insectary facility and upgraded it to a level 2 arthropod containment facility. And in Uganda, a level 2 arthropod containment facility was put up in 2019. And when you come to Ghana, in the University of Ghana, just earlier this year in February, a level 1 arthropod containment facility was inaugurated. This facility has the potential to be upgraded when the need arises to a level 2 containment facility. Next slide, please. And in terms of capacity building, team members are being prepared and trained on how to maintain modified mosquitoes using non-modified mosquitoes as a proxy. And so here, the team is using natural mosquitoes that have specific markers. And these specific markers are on the larvae, the immature stages of the mosquito. Some have red markings, others have white markings. And the team in Uganda are being trained on this to build their capacity such that when the time comes for them to handle the actual modified mosquitoes, they'll be able to use that same procedure and get the needed outcome. Next slide, please. And so with the monitoring of the facility, one of the biosafety measures is to ensure that no mosquito escapes. And as I did mention before, all these facilities are containment facilities, which are built to the highest standards to ensure that no mosquitoes get outside. And so any mosquitoes that escape from a cage within the lab itself are killed following protocols in the SOPs, in the standard operating procedures, where we have UV light traps. So these are traps that have UV lights in, and they attract the mosquito, any mosquito that escapes and kills them. And then we also have rackets, electric rackets that when they touch the mosquitoes, electrocutes them and kills them. And these are ways of ensuring that we do not have any mosquitoes that escape. We also have a double door system within all these facilities, such that when one door is opened, the other is closed, such that only one door can be opened at a time. And these are all measures put in place to ensure containment. And all data within this facility are kept in a database known as Earthscape, which is in the cloud, and that is a facility that ensures that those with authorization can have access to the data from these facilities. Next slide, please. And with our mosquitoes, they are all kept or reared under certain conditions of temperature and humidity. And there's the need for standardization. And so we have devices such as the Hobo, which are probes that picks up the temperature and humidity within these insectaries and kind of downloads them on a computer and into a database where we can monitor to make sure that the temperature and the humidity are within the desired ranges. And when they should exceed these limits, an alarm is triggered that makes sure that always measures are taken to keep the temperature and the humidity within the required ranges. Next slide, please. And when it comes to data management, we have specific ways of entering the data. We have specific forms that have been designed for use by all the labs, and data is input into the forms, which are then, as I did mention, stored in a database, and this database is the Escape. And then we do have a dashboard where the data stored in the database could be accessed by those with authorization to access the data. Next slide. And so these are all some of the biosafety measures that we do take to ensure that we operate in the with utmost care, and we ensure that all our activities are conducted to the highest standard of biosafety. Thank you all very much. As I bring my presentation to an end. Thank you. I'll hand over now to Divine.
Thank you very much, Dr. Fred. Thank you for the different attendants, also for their question that were popping during the different presentation. Now we are in a Q&A session, and I can see that Samuel has already raised his hand and he wants to directly ask his question. But as we do have some questions in the Q&A box, we'll start with them. And then after the different questions, we will open the floor, and then Samuel will be able to ask his question. So the first question I will take is from Esther Nakazi from Uganda, and I think Dr. Shalmugoya, this question is directed to you. She is asking, what is the level of risk that is acceptable to government? And also she do have a follow-up question, so I will give you both questions. And she also asked, do communities have to give consent? If so, how do they even understand the risk? So, Dr. Mugoya, you can answer the different questions.
Okay, thank you very much. It's a very interesting question. What's the level of risk that is acceptable to governments? I think to answer that question, one needs to understand what risk represents. And the decision makers sit in boardrooms every day to undertake analysis of risks which are acceptable to the people, on behalf of the people, or to governments. And they have a different methodology for doing that. In science, we also have a different methodology for understanding what's the level of acceptable risk. And we have even developed in science a code for scoring risks. So a risk can be low, very low risk. A risk can be high. In between, a risk can be moderate. And a risk can be totally unacceptable. So to answer that question, I think what governments do through discussion and through the process of consensus building, they're able to understand what level of risks are occasioned. Is it a risk to the economy? Is it a risk to people's health? Is it a risk to, so they determine, and this is done on a case-to-case basis, as I've said. Risks cannot be just addressed like that. Government has its own processes and procedures for evaluating the risk, but all that is based on consensus building, and it uses organs like parliament. Parliaments are bodies that governments use to do some kind of analysis. And within a parliament, you have a committee, a smaller group of people who can discuss and reach a conclusion whether the risks is unacceptable or it is acceptable. But that is only after the scientists, from the scientific point of view, have determined and characterized the risk as very low. Now, so in other words, governments normally would love to work with low things that have low risks, and they don't entertain any risks that is higher than low. So that's how I would answer it. It's on a case-to-case basis, and tolerable risks are those risks which are classified as low risks. Now, in the case of genetically modified mosquitoes for controlling malaria, I think the technology itself presents very low risks for application. So this is something that they can agree to go with. Thank you, Eric. And thanks, Nakazi.
Yeah, so when it comes to consent, yes, we do obtain consent from the community before we carry out any work at all within the communities. Additionally, we also get ethical clearance from the institutional review boards before we go out into the field. So yes, we do get community consent, and we do get individual consent as well.
Thank you,
Thank you.
Thank you, Dr. Fred and Dr. Charles. And talking about consent, in our next module, we talk about stakeholder engagement. So I think we have enough time during this module to discuss all the consent-related questions. Another question is still on the regulation, the regulation aspect. And this question comes from Mathieu Somda from Burkina Faso. The question is written in French, but I will translate it. So he's talking about the implication of stakeholder. And in Burkina Faso, for example, some organizations are against the use of GMO. So don't you yes, and for him, there is organizations that are against the use of GMO, like the study that Target Malaria is doing in Burkina Faso. So do you think that there are some explanation? So do you know maybe the reason why these organizations are against the use of GMO? The question is open to both of you, if whether Dr. Charles or Dr. Fred would like to answer.
Okay, I think it's very difficult to explain why those organizations are opposed to what Target Malaria is doing. But I can only, how do you say, conjecture and say that probably it's because anything that is unknown or in the African context is kind of feared, and people do not know much about what gene drives are, what genetic modifications are, and that is how we come in as scientists to try and use the scientific approach to allay all the fears that people have when it comes to genetic modification. And that is how come we have regulators who work with us to ensure that all that we do is transparent, it's open, and we follow due diligence throughout the entire process. So for us, we are trying to make sure that things are understood as much as possible to be very clear and transparent. And we hope that this will allay some of the fears, and some of these organizations will better understand the science and the rationale behind what we do to make it more acceptable to even those who oppose it. Thank you.
Thank you, Dr. Fred. I think your answer is very clear on that. And also I think the last question on regulation from Esther, who is asking how educated are African regulators to know about these technologies to be able to make good assessments. And she thinks that most of them know about plants, but not about animals. So how educated are African regulators?
Yes, I just want to put it in a slightly different context in order to answer this question. The question of education is important, but many regulators all over the world, there are people who have gone to school and they are educated, and they would understand some aspects of the science, but not everything about the science. So it calls upon, it's very important that the teams that are involved in the project, from the regulatory point of view, from the stakeholder engagement point of view, or from the communication point of view, they should work together to explain in simple terms the importance of the technology. They should also explain the rationale for the technology, the benefits and the risks. It's not a subject of education because these things are not learned in school. But at the end of the day, when we have a new technology, it's incumbent on us to reach out to these policymakers and take the assumption that they have enough understanding, to grasp the technical issues around the technology and be able to understand it. But I think at the back of our minds, the most important understanding comes from the burden of the disease. If our teams can explain the burden of the disease, to me, it appears like the burden of the disease, once you have understood it, it is easy to understand the technology that is being applied in order to resolve the burden. So I think our policymakers are well educated, but they need explanation. They need people who will go and explain the science and break the science down so that they can understand how the technology works to resolve their problem which they are raising. So it is very important that we do that. Yeah, thank you.
Thank you, Dr. Charles. Now the question regarding the ecology. So Mr. Sano from Burkina Faso is asking, currently, did you reach your initial objectives? If yes, what key findings did you discover? If not, what could be the reason?
Okay, so Thank you very much for your question. Yes, we are still in the research phase. We've collected samples for more than 3 years now. And from what we are seeing, we have not finished analyzing our data, I must say. So this is just a preliminary kind of output from what we've done. And what we are seeing is that when it comes to the ecological services such as pollination, it doesn't seem to, a kind of shows that Anopheles gambiae or malaria mosquitoes are not playing any important role when it comes to pollination. That's what our preliminary data is showing. And we do believe that conclusively at the end of it all, when all the analysis is completed, it will kind of affirm this preliminary results to conclude that Anopheles gambiae or malaria transmitting mosquitoes are not involved in pollination. In terms of their networks with other organisms within the adult niche and the larval niche, the data is being processed. And I would say that not until all the analysis is done, we can't come out and kind of say that they are involved in this or that, but all the indications are that they are not playing any significant or very important role within the ecosystem, such that if you significantly reduce their numbers, it's going to affect the entire ecosystem or the health of the ecosystem. It doesn't seem to show that. Our preliminary results don't show that.
Okay.
So
Thank you.
So that's what I can share for the time being.
Thank you, Dr. Fred. Also, another question from Esther before I give the floor to Samuel. Esther is asking the habitats or environments of all the countries where this project is happening are different. All countries do ecological studies. And also another question that she added, and she's just saying, mosquitoes are study subjects. Isn't it cruel to kill them using electrical rackets?
That's a very interesting question. Okay, so when it comes to the habitat, you realize that across West Africa, most of the habitats are shared. For example, we have the coastal savanna, we have the Guinea savanna, we have the Sahel savanna, and then we have the Sudan savanna before you go into the deserts, the dry areas. So most of these habitats cut across, but obviously there are minor differences from one country to the other. And the plan going forward, probably we may have to carry out similar studies elsewhere so that we have a broader picture of what is happening in the various habitats. But generally, if you take it on the general scale, it's basically the same. When it comes to being cruel to mosquitoes, I think this is a very debatable subject. Then we might as well not touch anything, because whether you are a vegan or a meat lover, there's always some killing of an organism. You either kill the plant before you eat, or you kill the animal before you eat. With mosquitoes, they transmit diseases to us. And in biological terms, it's like you kill or be killed. So if you don't get, if you don't deal with the mosquitoes will deal with you. So just think about that, and let's think about it to see if it is cruel or not cruel to kill the mosquito. Thank you.
Thank you, Dr. Fred, for this answer. Samuel, I've opened your mic, so you have the floor now. Thank you.
Okay, good evening, dear members on this webinar. Hope you're doing well and you can get me right.
Yes.
I have some few questions to put across to Dr. Fred and Dr. Charles Mugoya. One of them is that biblically, God created everything with a purpose, and we all know that. So saying that we are embarking on a move to kill an Anopheles mosquito, and we are contradicting to nature and God's creation. And two, referencing to what happened in China in the past years, where they resorted to kill all the sparrows because they were eating their grains, and then in the end result, pests attacked them, and then they actually smashed everything from their ground. And we see some effect coming out after killing this Anopheles mosquito out of the society and the ecology system. I will not go too deep into the discussion you said, Dr. Fred, on the mosquitoes and larvae being a meal to aquatic animals and pollination system. But there are some organizations, mainly on a criticism side, which are saying that yes, mosquitoes are of great importance in pollination. So I'm looking forward after making your research to get the results. What are your findings about this issue of pollination and being a meal to aquatic life? Two goes to, I'll go to Dr. Charles. Dr. Charles, yes, policies are policies, and there is no shortcut when it comes to policies. Uganda is up to date, not assented to the use of GMO bill. How sure are you that by the end of the research, the bill will be signed so that these GMOs are left free to be used in the country? And then we see Uganda, maybe many East African countries, yes, they are at good par, but take a look where Kenya bans Ugandan maize and other issues on some because of some issues. Don't you think that the bilateral understandings between these countries may come to contrary after the release of these GMOs? Because in Sase Islands, where I am, that is Kalangala district, you're doing your research on Jana Island. Jana Island might be 100 liters of fuel away from the border of Kenya and Uganda, meaning that by the release of these GMOs, they might go as far as entering into Kenya, and then this brings about misunderstandings. As we see in the business sector, they are not so good doing well. When it comes to science, what will happen? Have you already weighed the repercussions of it after all? And I want to finalize with one question. The community engagement. You've said that our next module will be on community engagement, but yes, Nakaze asked about the consent forms. I've done some few interviews where people were saying that, yes, we sign the papers, but we don't know what is embedded into them. Do you have qualified people on research who go to these committee members and specifically explain what is embedded in the consent forms that you give them? I thank you.
Okay, if you'll allow me to start, I think you have asked very relevant questions, which many people, incidentally, many people ask the same questions. And I think it is all there, I can take them all in the context of opinion, because as you can see from the questions you have asked, there's no recognition of the benefit of the technology. It is as if we are doing research for the sake of research. So where do the benefits of this technology come? Look at this picture. Malaria is spread by mosquitoes. And not all mosquitoes spread malaria. And Fred has ably demonstrated that only 3 species currently are involved in malaria transmission. And he has also told us that there are so many species out there, globally over 3,000 species, and in Africa about 80 species are roaming, and they are within us. They are also biting us, but they are not transmitting malaria. So we are dealing with 3, and these are the 3 which are causing the havoc that we are talking about. Now, Fred has also said that the aim of this technology is not to wipe out a species from the earth. The aim is to suppress the population or to replace it. So that means that we are only interfering to the extent that the numbers of these mosquitoes will be so low as not to transmit malaria, because extinction is not part of what we are trying to do here. And then from ecological studies, those who have done science of ecology know that species can be separated by barriers, environmental barriers. Like you could have a desert where a species from one area cannot cross to the other one, or an ocean, or a lake. So those natural barriers exist, and that's why if you look at Africa, the distribution of mosquitoes is not even. Some areas have more than others. And therefore, that's why our approach is on a case-to-case basis. We are identifying those hotspots where we can use this technology to suppress the species that cause malaria. So our purpose is not to create total eradication. Then on the issue of the bill in Uganda, the Genetically Modified Act, or Genetically Engineering Act, is supposed to regulate how these organisms are supposed to be released into the environment. Currently, we are using other laws to be able to do studies in the laboratory. And we need those studies in the laboratory to give us a way forward when we release them into the environment. So when the Genetic Engineering Act is passed by Parliament, it will give us the guidance that we need for the implementation of the release.
I think we lost Dr. Charles.
That will help us, yes. Yes,
Okay,
I'm finishing, yes. We have enough laws to do the studies in the lab. And then on the question of the geopolitical nature of our being in a regional setting, what's happening in Kenya affects what's happening in Uganda. That's when I said that I think we need a regional approach, a regional conversation will be very helpful as we develop this public good. The public good is the genetically modified mosquito that can help to suppress malaria in areas where malaria is a problem. I think that I'll stop there for the time being, and if Samuel can contact me, I can give you more details. Eric?
Thank you, Dr.
Yeah.
Shah. Yes, Dr. Fred, if you want to add.
Yeah, just a quick comment. So Samuel asked if we are not playing God. I think it takes us back to what Charles said earlier. It's about the risks and the benefits. Let's look at the burden involved in malaria and decide whether to leave the situation as it is without any intervention, or if we think that the burden is huge enough and we have to do something about it, then there is need for us to do what we are doing now. And then Samuel, you also asked about, you used the Chinese experience of swallows, killing of swallows as a possible ecological outcome. Yes, that is the more reason why we are carrying our studies here, to better understand if these mosquitoes play any role within the ecosystem at all. And so I think when the time comes, when all our data is ready, we'll be in a very good position to tell whether significantly reducing the numbers of these mosquitoes is going to have no effect whatsoever to the ecosystem or some effect. So I think very soon our results will be out there to answer this. And then, yes, some mosquitoes have been reported to be pollinators, but you realize that all those reports come from the temperate regions in the Americas and Europe. But we don't have any information on mosquitoes involved in pollination in the tropical world, in our part of the world here. And that's why our study is very important. And from all indications, they are not playing a very important role. Yeah. Thank you.
Thank you very much, Dr. Fred and Dr. Charles. And I'd like to remind our attendants that Dr. Fred and Dr. Charles are available for interviews or for follow-up questions. So if you do have questions, you can just email me and I will liaise with the different team in order to set up the interview with the relevant doctor. Dear doctors, I will just give you the floor for your concluding remarks, and then we will close today's training session. So I will start with Dr. Charles and then end with Dr. Fred. You have the floor, please.
Okay, thank you very much. And we thank all the participants who have been able to participate in the Health Journalism Bootcamp. I think the purpose of this bootcamp is to share information and also to clarify on some of those areas which are not clear. And speaking about regulatory environment, I think we all recognize that it's a very important element of the development pathway. We need regulations because we cannot work in an environment which is a vacuum. Now, the importance of regulation is that they give us certain benchmarks, they give us certain standards, and they define the scope in which we operate. And we have agreed as a project, Target Malaria, that we will obey and comply with all the benchmarks that have been sent by regulatory bodies in the countries where we work. So this is our pledge, that we will comply with all the laws and regulations, because the laws and regulations are aimed at protecting the people and the environment. So this is our pledge, and we will not work outside those laws and the regulations that are available. And that's why we consult and we work with the regulatory bodies in order to achieve our objective. So, yeah, this is the message that I would like to air out, so that we are all on the same page as far as this one is concerned. Thank you very much.
Thank you, Dr. Charles. Dr. Franz? Yeah.
Yes, I will just want to add a little to what Charles just said, and say that it's a great opportunity for us to meet.
Maybe we can come back to this.
Yeah, yes, I will say that it's a good opportunity for us to meet and talk about these things. So I look forward to a future where we will have more of these interactions, where we can share ideas to kind of better understand this technology, to kind of better understand the risks and the benefits associated with this technology, all for the benefit of all of us and humanity as a whole. So thank you all for making time to be part of this, and I look forward to a future where we can all work together for the benefits, common good of all of us. Thank you.
Thank you very much, Dr. Fred and Dr. Charles, for your time and for sharing this information with us. Thank you, Davin, for co-moderating the session with me. It has been a pleasure for us to host this session and to have you sharing this different information with us. We remain available should you have questions, or would you like to have an interview with one of our speakers. In the meantime, you can follow Target Malaria social media account to have all the different information. Our next session will be held Tuesday, and it will be about stakeholder engagement and a community agreement model used by Target Malaria. I will give you more info in the different emails, and I wish you all a good day. Thank you very much. Bye-bye.
Thank you.
Thank you.
Bye-bye to you all.
Bye.